So Rich, you have the first question.
Oh, thank you, Constantine. So my question is for Dr. Petrick as soon as he gets settled. And I'm over here. I was quite glad to see, I'm over here. I was quite glad, I'm Rich Shilsky from ASCO. I was quite glad to see the sort of evolving paradigm as you know, software as a medical device. And I'm just sort of curious as to where you are in your thinking about that with respect to whether software that's intended to be used in this broad area of clinical decision support would be subject to regulatory oversight. So, you know, if you follow the kind of a paradigm that Chris Kogel laid out this morning, you know, I have a software algorithm where the input is a, you know, multiplex genomic test, and the output is possible drug treatments for a patient with cancer, but it's up to the discretion of the treating physician, you know, whether to accept those recommendations or not, and if there are multiple options, which one to use. Does that kind of usage of software fall in the paradigm that you're thinking about?
So I would say most likely that example likely would be a device that we'd look for data on to support. You know, this is still sort of evolving in what it means to have informational, you know, what is informational relative to a diagnostic or treatment tool? I think that's a little bit of a hard to define completely, but the idea of when you're giving diagnosis or you have a strong impact on the diagnosis, even if the device itself isn't working completely independently, but in working in conjunction with the clinician, that type of analysis would be something that would be regulated. And then the question is, what type of data is going to be needed for that? And I think that's the type of question of what we're trying to work through is what type of data and under what scenarios. And so we're also dealing with this question of algorithms that change. So we can talk about fixed algorithms. They're fixed. They go to the market. There's also this whole pathway in AI and machine learning we'll talk about specifically, where there's the ability that these algorithms can utilize data in the clinical setting, and how would we go about doing that? And it's another question that we don't have any great answers for yet, but we're trying to work on and certainly open to ideas and suggestions of how to deal with those types of paradigms to make it efficient, but also to have enough of a level of validation that we're comfortable with how they work. The other area I'll just say is complicated in this whole scenario is what information is getting back to the clinician. So if you have these algorithms that change over time, which is maybe great, maybe they're working even better, does a clinician know that? If I'm sitting here, if I have an algorithm that's, you know, say specific for California, because that population has one population and it's different from New York, what happens if my clinician moves from New York to California? How are we going to deal with those types of scenarios? So I think there's a lot of these sort of other aspects that are beyond just does the device work in of itself, but how is it going to be implemented in clinical practice that are somewhat to do with the regulatory side of things, but also much more larger questions about how these things could work and how they might work. I think those are great
questions. You know, I just, you know, if give me one more second. You know, I think if you look at the way, I'll only speak for oncologists since I am one. If you look at the way many oncologists look at, you know, this kind of information, they're really not that concerned about what's in the black box or how it works. I mean, you know, the issue is, you know, what's the clinical context that I'm dealing with, and does the output from the black box help me make a better decision? And whether or not the output is actually even relevant to their decision-making process, I think is oftentimes not something that's well considered. But it's going to get to the issue of, you know, what are the data required to support both regulatory decision-making and clinical adoption, and you know, what's the level of evidence that's necessary?
Thank you. Yes, from here.
I'm Türkan Gardiner, applied statistician. And thank you, by the way, for this fascinating session. I was taking notes all the time. A couple of comments relative to Dr. McShay's presentation, and these are provided as a supplementary generalizations that I have used from multivariate analyses over time. Your first comment in terms of allowing enough degrees of freedom and not oversaturating the models, that is definitely a problem in multivariate analysis. The rule of thumb I have used, and some of my colleagues, is that you allow 1/3 for estimating error and 2/3 of the degrees for example, if you take n as the rows, as the number of rows, and the y-axis for the parameters to be estimated, you use a 1/3 to 2/3 ratio. Don't try to evaluate any more than 2/3 of the number of samples that you have collected. The second is a research that exists in the literature called intervention analysis for time series. For example, if your reagents change or the structure of data collection changes, as I have seen in surveys, there is methodology for intervention adjustment. The Box-Jenkins-Chow time series analysis allows for that. You model pre-intervention, and then you do a modeling for post-intervention and adjust the regression coefficients that are obtained. And this is fascinating because when I was with helping Energy Information Administration, we ran into that. And when your intervention time is known, you can do the 2 sets of modeling and adjust wherever necessary.
Thanks.
Thank you very much for your presentation.
Thanks. Lisa, do you have any comments?
Just quickly follow up with one comment. I think you're making the point there are some methods already out there, even for sort of standard multivariate regression.
Right.
And I think that one of the reasons we've had some difficulties with taking machine learning methods into things like omics is that most of those were really developed for situations where you had lots and lots of independent subjects and you might have a lot of variables on each subject. And even in our biggest studies in omics, most of them are not more than a few hundred patients, and we might have 10,000 or more measurements on each patient. So we just have to be careful not to take leaps too quickly from what we're more familiar with to these other situations.
Thank you.
Thanks.
Yeah, great presentations, Chris Kogel. I'd like to force a question borrowing from the drug development paradigm. This is for everyone. In the drug development paradigm, we have the FDA, as Sean Cozen presented, has a pretty clear development process, and the FDA label is quite explicit on what the drug is, what its indications are, what its mechanism of action is, what its exact indications are. Should that drug development regulatory paradigm, should it be applied to software? And the reason why I'm thinking of that is someone could say that a medical device that interprets data that leads to a treatment decision with high risk, like most chemotherapies and targeted therapies, should be in the hands of physicians and maybe not in the general public. And secondly, the reason why the FDA has the market label is so that there's transparency. And so to Pratik Shah, would software developers be comfortable putting portions of their code in a prescriber's label? Should we, to the FDA, should we limit the use of some of these SAMDs, these software as medical devices, to physician-only use and not to the public?
Yeah, I'll take it quickly. Thanks. I think as a person who develops software, yes, you should make your code publicly available. Yes, you should make your data publicly available. You should make sure that there were no biases in your data. And yeah, that sounds good research. Yeah, sure. I think we worry in computer science when regulation comes from a different field, which is more on deterministic medicine, one molecule, one drug, while Some of the algorithmic work we do is slightly more liberating in a different way, where many outcomes are possible from the same data. So for a physician use, a label is recommended, but maybe not for wider interpretation of the algorithm and the code. That would be my take.
So just from a regulatory, we do have labeling, we do have indications for use, we have devices that are specific for clinical use, we have devices more generally open for, so all those questions you had about label, those exist. The way studies are done may be different and certainly more flexible in the device framework. One of the reasons is we just have thousands and thousands of devices that we deal with across the whole spectrum. So when we're talking about SAMD, we can't talk about it as a monolithic, there's a whole variety of different types of devices that may have very low risk associated with them, so our approach is a little bit different. And so from that perspective, it's very different. But those basic ideas of labeling and indications for use and how they're utilized and how you can market, those all exist for devices as well as for drugs.
Ed, can I ask a question to you?
Sure.
To what extent, because drugs and devices have different statutory rules about degrees of evidence and proof, do we need that to be different to get a system that would be optimal? Or is there enough both latitude and restriction within the current system to do it right?
So I'm not going to comment exactly about that. That's more of a congressional. Congress has gone and passed a new 21st Century Cure bill, and we live within the regulatory and the legal framework of what we have from the Congress on the laws. Could there be better? Certainly, there could be things that could change that would be better. I think it's this balance, again, of being what is the right flexibility relative to devices or certain classes of devices relative to others. And we have our own opinions internally, but Congress gets to weigh in and develop on what are the actual laws that we need to follow. And some things are less than ideal because the framework is put in place for other reasons beyond just what we think is best.
So we'll take one more question from the FDA then.
Sean Cozen, FDA. Very interesting presentations. It seems like we're facing as we move into a world where we have a lot of data to profile patients genomically, proteomically, and we have a lot of data points that allow us to target our therapies. So we know a lot more about the potential of the therapy that we take to the clinic to have an impact in drug development. The old days of basically just throwing something out there to see if it sticks, do a randomized study are over. So we're much more intelligent in how we design drugs and take them to the clinic. So it's starting to pose a very interesting dilemma where the math that we use to assess the safety and effectiveness of therapies are based on stochastic principles of causation. Meaning that things just could happen randomly. If you give a drug to someone, tumor shrinks, could have been due to chance. Whereas we know biology is not stochastic, it's deterministic. So as we move, so do you see an opportunity to bring new math into the equation? As we have more predictive capabilities with AI, other genomic data sets, and other data sets, to start thinking about, deterministic causation, which is really what we're facing now. And this is starting to have real impact. For example, if a clinical study, a randomized, let's say, clinical study, hypothetical study, that doesn't meet its endpoint, however, statistically, meaning, they don't meet that p-value margin. However, clearly, we see this all the time. There are patients who respond. There are tumors that shrink. They may be outliers, but it's happening. Those innovations, those drugs can be shelved. And that may be doing disservice to the patient, at least the responders. So by combining deterministic causation, a new math basically, with adaptive clinical trials, do you think there may be an opportunity to be more targeted as the drugs that we're developing and how we actually generate the evidence?
Are you saying that we're in an era where there could be 100% response if you know enough detail? Is that what you're saying?
Well, if you have one response out of 1,000, most likely, especially in oncology, that response is real. It's not due to chance. Tumors don't shrink just by chance. But if you don't meet your statistical boundaries in that study, those few responses won't matter. Because the drug can still be shelved because it would be deemed unsuccessful. So there are a lot of failed studies that have had subgroups that have responded. And those subgroups are essentially thought that, since the study didn't meet its p-value, then it could have been due to chance because the statistical principles, the underlying principles, are based on stochastic causation, not deterministic, causation. However, smart clinical developers that are able to pivot, study those subgroups, those exceptional responders, we call them, and redesign the clinical studies to target, those exceptional responders. So they enrich their clinical studies to only include those exceptional responders. Yeah. But not every developer has the luxury of going back and extracting those exceptional responders, enriching another study to include them. Gefedimab is a great study in the days of a great use case, days of EGFR mutation. We didn't know about the mutation. The study showed promise, but it was only being driven by EGFR positive patients. So it's interesting to think about, is there an opportunity to bring new math into how we determine causation when we look at things empirically.
Give us a chance here. I think Pratik wanted to say something.
I just want to make comment on Sean's point.
Steve, Giovanni, did you want to say something too on this? This is kind of, it's a big one. We have so many
I'll
things.
say a small thing,
100%.
and then you say the big thing.
Yeah, please go ahead.
So very quickly, we have 1 minute, 16 seconds.
No, don't worry about it.
Okay.
Take 14
so I think
seconds.
so Sean, you make a great point, and I've been talking to adaptive trial, as you know, pragmatic clinical trial people, right? And they have these arm assignment strategies where they calculate a hazard ratio for a certain patient. That hazard ratio is mostly on Kaplan-Meier curves. Boom, you're assigned, right? And then after 5 or 10 patients, they gauge the response and they start the arm. Most of that work is not data-driven, or the point you're making is it's data-driven with statistics, which you don't like. It should be data-driven with logic and common sense, right? So I think to the statisticians in the room, this is an open question. How do we introduce logic in non-data-driven equations which are beneficial to the patients?
Steve will answer.
I'll just make one comment. Just because we use probability doesn't mean we're inferring that the process is stochastic. Every single cancer patient we treat today, it could be deterministic. The probability can be reinterpreted as our uncertainty about what's going to happen, right, even for deterministic processes, of which maybe everything's deterministic. Right. So I don't know exactly. I mean, everybody would agree if we could identify the factors that predicted probabilities near 1 or 0, the subgroups, we'd want to do it. And you can call that causation, but the probability of something happen doesn't necessarily mean that it's happening by chance. It's just that we don't know. So I don't know precisely what math you're talking about. I do think that your perspective, that the possibility that every fate is determined, and our getting closer to understanding what that fate is, particularly looking at heterogeneity among subgroups and understanding the complexity of biology, are necessarily discordant. Maybe Giovanni has a different perspective.
Giovanni has the last comment, and tomorrow you could bring this up again. That's why I was asking whether I've never seen 100% subset response. So,
this doesn't have to be 100%.
Well, to be deterministic, you have to get to that point. In other words, you have to take out the uncertainty. If you don't have the uncertainty, then if you have the uncertainty, then you're not deterministic.
Right.
But
just as a quick
Giovanni
comment
has
about
the
that.
point.
So no drug is 100% effective, but that's the risk that we take. We know that the number needed to treat is pretty high in a lot of cases because a lot of patients actually don't benefit. So
it's not deterministic. There's still a probability, it's just probability is very
right,
high.
absolutely. Okay. So this is about not abandoning a potentially useful intervention just because our statistical boundaries under those certain conditions are not met. So that's where adaptive, clinical study designs that are more logical can come in.
Giovanni.
I think we're on the same page in terms of the general concept that a substantially increased degree of adaptivity is what's needed to come to much smaller strata that are much more homogeneous and where the level of prediction is much more accurate. As Steve is saying, it will it will be difficult to disentangle the part of probability that represents the scientific community lack of knowledge about what will happen to specific patients in the subgroup from some intrinsic stochasticity that I think we'll always need to deal with in oncology. I mean, if you look at the simplest model, which is TKI resistant to patients who have BCR-ABL mutations, those are accumulated by mutagenesis, and the timing of that, or even whether that occurs or not, is subject to the fidelity of duplication of those cells, and it is to some extent random for within the history of an individual patient. The denominator to the randomness is the number of cell divisions there. So I think there is a core of stochasticity that even if we could agree entirely on the knowledge part and we didn't need probability to describe that, will still be something we need to grab. But in terms of the big picture, I think we're on the same page.
Thanks. That's the last comment. We can take this all up tomorrow afternoon. There is a panel and so on. Let's thank the speakers. And the reception is on the 3rd floor of the building. And it's starting right away, and the drinks are up.